I hope you see the slides from my display. The title of the webinar is Release of the Nuclear Medicine Patient in Europe, from basics to a computational approach. This is a joint EURADOS-EANM initiative. First, I will give you a very short introduction on EURADOS. EURADOS means European Radiation Dosimetry Group. It is registered in the German Register of Society of a non-profit association. It is composed by 4 main bodies: the General Assembly, the Executive Board, the Council, and also several working groups. EURADOS is a network of 83 European institutions, including the voting members and more than 650 scientists as associate members. The financial resources originally come from the sponsoring institutions, from voting members, and sometimes from the projects funded by EC, etc. EURADOS have organized every year annual meetings. The next will be held in Bucharest, Romania, the February 24th to 27th. Second, I will also give a very short introduction to the pilot group. In 2020, the EURADOS update the strategic research agenda and put the dosimetry of patients and the staffs in nuclear medicine in the agenda. And 2021, it is proposed to set up a working group in the nuclear medicine dosimetry in the annual meeting 2021. The council announced the establishment of the pilot group in dosimetry nuclear medicine in Belgrade annual meeting 2022. We have several ongoing tasks. For example, the first one, EURADOS-EANM joint project on the external dose rate, led by Lara Stulens. We have also another project like AI in the nuclear medicine imaging and also dosimetry. We have some publications, for example, and also EURADOS report. We pilot group also participated in the tactic grant challenges of American Association Fixes in Medicine in 2023 and obtained the 3rd position. Pilot group works very closely with EAAM, Radiation Protection Committee, as well Dosimetry Committee. We work closely with IFON user group Siegfried, and of course with ICRP. The next pilot group meeting will be held virtually in December, the 5th December. If you like to join, you can send me an email and I will give the links. Now I just introduced the talks of the webinar, because this webinar is a joint action from the EAAM and EURADOS. Therefore, I introduced the chair of the Radiation Protection Committee, Jens Kurt, to have a short introduction. And second is will be introduced by Peter Covens on the legislative aspect of the patient's release in Europe and focus on the results of the Simple Red project. The second speaker is Christoph Müller, is a professor from the, will give the clinical experience and needs in the radiopharmaceutical therapy. And the third one will be given by Lara Stulens, computational approaches to access external dose rate from nuclear medicine patient. In the last session, we will give a read out the questions and give your answer. Yes. And now let's go to enjoy the webinars. I think the first is Jens Kort. Jens Kort is chair of the EANM Radiation Protection Committee and also the chief medical physicist at the Department of Nuclear Medicine at Rostock University. Please, Jens, the floor is yours.
Okay, thank you very much, Weibo.
I will close my, yes.
Okay, so I will try to share my screen.
Yes.
Okay, so I think you can see the title slide. Okay, this looks, sorry about that. Come on. Okay, so let's start anyhow. Yeah, thank you, Weibo, for the kind introduction, and I would also like to thank the organizers of EURADOS for putting together this webinar on a very important topic that is currently occupying all of us very intensively. And also thank you very much for giving me the chance to introduce the Radiation Protection Committee of the EANM a little bit. We are currently 5 members from left to right. This is Yannis Iakovou, physician, nuclear medicine physician from Greece, Uta Eberlein from Germany, medical physicist, also Elena from Spain, medical physicist, Sigrid Leitz-Weckborn from Malmö in Sweden, medical physicist, and this is me, also a physicist. So we are more on the physics side at the moment, but nevertheless, as we are all involved in a clinical routine, we also have the clinical needs a little bit in mind. Among many other things one does as a committee member, an important task of our committee is to connect the EANM with a variety of research platforms and organizations dedicated to radiation protection medicine, both the European research platforms and also societies on the European and on the international level. Like the other committees, the Radiation Protection Committee is also involved in the preparation of the annual Congress. The same applies to webinars and on-site seminars of the European School of Multimodality Imaging and Therapy. And in addition, the committee members are also involved in the EANM radiobiology initiative, for example, to raise awareness of this important topic. Oh, come on. Okay. And the first paper on this topic was published just a few days ago. Furthermore, the committee is currently working on 3 guidelines. The first guideline on extravasation is being developed in close collaboration with EURADOS. And the aim is to compile the current knowledge on extravasation and diagnostics and therapeutics, and to provide clinical active colleagues with a guide on how to proceed when extravasation occurs. In particular, the topic of dosimetry in the event of an extravasation is repeatedly the subject of controversial discussion. Also, the topic of breastfeeding after the administration of radiopharmaceuticals is asked about again and again, and we are compiling recommendations on the basis on the currently available data. We know, and you are also aware that at the same time ICRP is working on the revision of their corresponding recommendations. And Sigrid Leitz-Weckborn is also involved in the ICRP. And as she is also a member of the Radiation Protection Committee, we have very close links here, which is very important at this point. And finally, we are also working on a guidelines for radiation protection aspects in lutetium-177 therapies that will most probably, or we'll try to cover the topic as broadly as possible, including aspects such as production, application to patients, and etc. And also, of course, the issue of outpatient or inpatient radionuclide therapy is an important one. And as you are most probably aware, the regulations in Germany are very strict. So here in Germany, we have to perform all radionuclide therapy on an inpatient basis. So I'm somewhat biased, and therefore I'm also very much looking forward to the upcoming talks, as we need to replace this, or we need to place the sometimes controversial discussions on a sound and scientific footing. So also a note on our own behalf. So if you're interested in working with the EANM, there are several opportunities to do so in a wide variety of areas, clinical, but also on the physical side, on the physics side. In addition to the various committees we have at the EANM, there are also projects or task groups, for example, of all which makes an important contributions to specific topics in nuclear medicine community. So if you are interested, please feel free to register and as an interested volunteer on the respective EANM website. And finally, I would also like to draw your attention to an important EANM initiative that relates to the workforce crisis that drives us all. Nuclear medicine, as you know, is a very vibrant field with a high degree of multidisciplinarity. And in order to attract young people from all the professional groups involved for a career in nuclear medicine, also in the future, the EANM has launched a program specifically for this. And the aim is to provide a platform for networking, involve young professionals into activities of the society, and to organize events and sessions on how to build a career, and many, many more. So if you are interested, you can find more information on the EANM website, and of course also on the EANM LinkedIn channel or the other social media. So thank you very much for your attention.
Thank you very much for your introduction on the EANM Radiation Protection Committee. Jens, very good. I think people will contact you if they like. And now we start our scientific presentation. The first is the Peter Covens. Peter is the head of the research department radiation protection in the University Brussels. He is also former member of EANM Radiation Protection, as well as a member of the pilot group and also the working group 12. Peter, please, the floor is yours.
Thank you, Weibo. Hello, everybody. I would like to talk about the legislative aspects related to the release of the nuclear medicine patient in Europe. First of all, why do we perform a release process? Well, first of all, the release process aligns, in fact, with the optimization of radiation protection, and we try to ensure that the radiation dose to individuals is minimized. But there's one thing we may not forget, that in fact also if we have a good release process, it allows also the patient to return to normal life as soon as possible. So in fact, it's a balance. It's a balance between beneficence and non-maleficence. We know that we have medical benefits of the therapeutic or diagnostic procedure of the patient, but by controlling when and how the patient is released, we also have the risk of harm to the patient, their family, and the public is minimized. And for the harm to the patient, I don't mean here the radiation burning, but it's mainly the level of restrictions that does harm to the patient in this case. So what is written in the Euratom Basic Safety Standards Directives? When we go first to the origin, it said that member states shall ensure that those constraints are established for occupation exposure, for public exposure, and also for medical exposure, and this relates to the protection of carers, comforters, and volunteers. This is all in the framework of optimizing radiation protection. And those dose constraints, if they are established, they should be established on the individual effective or equivalent doses. And they also should be defined using an appropriate time period. Furthermore, which is related to the dose range for the public exposure and also the exposure of comforters and carers, we have a need for adequate release criteria of patients from the hospitals, together with the provision of instructions. And when we go deeper, it's also stated that member states shall ensure that in case of a patient ongoing a treatment or diagnosis with radionuclides, the patient or representative should be provided with information of risk. And together we should also provide appropriate instructions to restrict the doses to the persons in contact, not a LARA here, but it's stated here, as far as reasonably achievable. And for therapeutic procedures, those instructions should be written and they should be handed out before leaving the hospital. So over the years, a lot of recommendations and reports handled the issue of either dose constraints and release criteria or dose constraints and release criteria together. But what we see today, we still see a lot of, or a very high level of heterogeneity across Europe. Heterogeneity of using dose constraints, using release criteria, and using release instructions or patient's instructions. And even within the same country, we can see regional differences. And we can see even differences within the same hospital when it relates to non-written instructions for diagnostic procedures. And that heterogeneity was also confirmed during the SimpleRAD project. So this SimpleRAD project was a SAMIRA study on the implementation of the Euratom and EU legal basis with respect to the therapeutic uses of radiopharmaceuticals. And it ran during 2 years, between 2022 and 2024. The final report of the SimpleRAD project will be published very soon, within a few weeks. But today I will give you some results that are in fact related to the release of the nuclear medicine patient. So what were the SimpleRAD objectives? We try to understand, in fact, in this project, the links and the interdependencies between the European pharma regulations and, in fact, the Euratom radiation protection requirements laid down in the Euratom Basic Safety Standard Directive. So we try to propose a practical guidance, and also we try to explore the quality and radiation safety issues related to several types, namely the current use and introduction of novel therapeutic radiopharmaceuticals, the implementation of patient dosimetry, the role of the medical physics experts, the management of radioactive waste, and the subject of today, also the release of patients from the hospital. And we used a method consisting of four work packages. The first one was rather an analysis of the interrelations between both legislations. And the second one was an interesting because we performed there a survey on the implementation of all European legal requirements among stakeholders. And in the third work packages, we recommend actions to advance in the future on the coherent implementation of all European legal requirements. And at the end, we organized also a workshop some months ago. So as part of the work package 2, the survey among stakeholders, we asked several questions related to the dose constraints and release criteria. And our stakeholders were health professionals, competent authorities, national societies, national training bodies, and even the industry. And with a special question or dedicated questionnaire, we received almost 200 valid responses. No, so let me go through the questions related to the constraints for comforters, carriers, and release criteria of patients. A very simple first question we asked to the stakeholders is, are you familiar with the concept of the dose constraints for comforters and carriers, and also the public laid down in the Euratom Basic Safety Standard Directive? And surprisingly, more than 70% of the respondents answered yes. But when we went a little bit further and asked, can you provide further information regarding the use of the constraints, we get quite a variety of answers. Some answered with, yes, we have one. It's a numerical value of the effective dose that we use as a constraint. Others said, yeah, we use the numerical effective dose value of the public dose limit for both comforters and carers and the public. Other answers were, no, we don't have that. It's a decision of the practitioner. And some of them didn't really understand the question or were not familiar. Instead, they said they were familiar. And they answered with the release criterion, can be in days of hours, or even a dose rate, a measured dose rate value as that they use as a constraint. And other respondents also referred to the national regulations. So when we went again a little bit further and we asked, well, what is then that numerical effective dose value of the dose constraints? We got again a variety of answers. Some of them don't know, we don't have there is a value. They said they had a value, but there's no specific numerical value of the dose constraints. And other really responded in detail by specifying a numerical value. And they also made the difference between public, comforters, and carers. And within the latter group, they also said there are several dose constraints and they are age dependent. But what we also can see, and what was laid down in the Euratom Basic Safety Standards, is the appropriate time periods. And this was translated either into per year, or sometimes per treatment cycle, or sometimes per single procedure. So related to this, of course, to the dose constraints, we asked also if there was guidance provided concerning patient instructions and release criteria for specific therapeutic procedures. And this was the case in for half of the respondents. And as an example, as illustration, I give here some specific release criteria. There are more for other radiopharmaceuticals in the full report, but just for illustration, I give here for 7 countries the specific release criteria of the country for lutetium dotatate and lutetium PSMA. And what we see, depending on the country, sometimes there is a very strict release criterion, or sometimes there is no release criterion, and people often use time post-administration as simple release criterion, and sometimes measured dose rates at 1 meter, or sometimes it's a combination of the time post-administration and the measured dose rate at 1 meter. So this simple rat survey said again, or highlighted again, once more the heterogeneity of both dose constraints and release criteria for nuclear medicine patients. And what was also seen is that the responsibility of setting these constraints or release criteria varies across the member states. Sometimes they were established by competent authority, sometimes by professional bodies or societies, and sometimes by the treating centers themselves. But when we look deeper, we also saw that different criteria and decision levels can influence, in fact, the final settings of those constraints and release criteria. In fact, if we want to protect an individual of a critical group by using patient release criteria and patient instructions as final result, we have to consider that the process of setting this release criteria is influenced, in fact, by different criteria and decision levels. And the first level is a type of exposure that should be considered for an individual, in fact, that might be exposed by a nuclear medicine patient. If the individual acts like a comforter or carer, this exposure should be, in fact, considered as medical exposure. And we have seen in simple rat that there's an absence of specific dose constraints for, in fact, the medical exposure, this type of exposure, in many countries. And in fact, that there's a need of guidance that will help individuals in the decision, in fact, which individual and in which situation this individual can be considered as either a comforter carer or a member of the public. The second level is in fact the decision on the level of the dose constraints. Those constraints are set in fact not as a limit, and they do not represent a demarcation between what is safe and what is dangerous, but they are used as an optimization of the radiation exposure, and they are used as a prospective tool in this optimization. We have to say that for planned exposures that have in fact a dose limit associated with them, like the public exposure, those constraints should be in fact set at a lower level. And then we have the third level that has in fact an impact, and that is in fact the used risk assessment method. Different in fact implied methods in the past can result in fact in different patient risk criteria and instructions. And within the fact the increasing number of therapies, a better knowledge of patient biokinetics should be done, and we should encourage, in fact, risk assessment studies using state-of-the-art methods. And this is an example that is done, in fact, in the EURADOS-EANM joint initiative, where biokinetics, where there's improving going on of biokinetic models, and there's a more detailed calculation going on for external dose rates for close contact scenarios, not using the inverse scroll law after dose rates measurements at 1 meter. In fact, patient release criteria are often based on dose rates measurements at 1 meter. And if you try to have some risk assessment studies during specific exposure scenarios like close contacts, the error you made by using the inverse square law after dose rate measurements at 1 meter is very high. But it will be better explained by Lara Struens today in the third presentation. So I also share one or some of Simperat proposed potential remedies, and one of them to go to a further harmonization level, in fact, and one of them is, in fact, the generation of data. Maybe we should think about installing an EU database of measured doses for family members and comforters and carers, and also establishing a database of dose rates can be measured or even calculated, and exclusion factors, including biokinetic data. And we should think about guidance documents that will help in the decision of which individuals and in which situations those individuals can or should be considered as a comforter or carer, on the other hand, as a member of the public. And we have seen also, because a lot of respondents answered very weird things, that we need to have a guidance document that explains the differentiations between limits and constraints, and that also can propose constraints for the public and comforters and carers for nuclear medicine procedures. However, this should involve national competent authorities and also HERCA. To conclude, we have seen that the release process of nuclear medicine patients following both therapeutic and diagnostic procedures still vary widely, not only across Europe, but also across the world. And Simperat confirmed this finding, but they also, within the project, we also identified factors and proposed potential remedies. And it's a fact that all the different initiatives that help us to go towards a higher level of harmonizations should be carried out at different decision levels, which could help us to go towards this higher level of harmonization. And with this final remark, I would like to thank you.
Thank you very much, Peter, for the excellent introduction for the registration for the release of the patients in Europe. Yes, yes. If you have questions, you can write down in the chat, and in the end, our organizer Francesco Kesting and Agnieszka will read out and answer you together. Our next excellent speaker is the Professor Christoph Müller. Professor Müller is the professor at Nuclear Medicine Department of the University of Brussels, as well in the academic clinics Delta in Rosalia. Professor Müller was EAAM president in 2017 and 2018. Christoph, the floor is yours, please.
Okay, thank you. You see my slides? No, not yet. Okay, now it should work. Thank you for this opportunity to speak about the clinical experience. Oh, what's happening here? Just a moment. To speak about the clinical experience and needs in radiopharmaceutical therapy. So, my task is to specifically focus on the release of nuclear medicine patients in Europe. And as we saw in the previous presentation, there is a large heterogeneity. So diagnostic and therapeutic applications in nuclear medicine are inherently related to radiation exposure, not only to the patients, but also to the health workers and the public. And obviously, the radiation exposure should be kept as low as reasonably achievable by justification and optimization of the procedure, training and education of the health workers, and an adapted communication with the patients and their relatives. However, nuclear medicine also covers, besides radiation protection, a lot of other aspects. Such as the potential clinical benefit, diagnostic and therapeutic alternatives, the prognosis, the adverse events, and the quality of life, as well as the importance to ensure global access to new diagnostic approaches. And when we talk about nuclear medicine, there are two exposure pathways. On the one hand, the external exposure risk, which is always present and typically is in between 5 to 20 microsievert per hour at 1 meter of the patient. And on the other hand, the risk of contamination, which should be avoided through hygienic measures. I will mainly focus on the external exposure risk in this talk. So how do we get an idea of the exposure to the public? But there are not that much data available, but there are quite some data available when it comes to occupational radiation exposure in nuclear medicine. And this slide is just to show that we are really talking about other levels of dose rates compared to radiology. So in radiology, a lot of health workers are exposed to quite relatively high doses compared to nuclear medicine when they're next to radiology or X-ray apparatus, while when they are a nuclear medicine patient, is also a source of radiation, but a source of radiation that is well below the dose rates known with X-rays with 2 orders of magnitude. So okay. So in order to have a view on the dose rates in occupational exposures in nuclear medicine, I would like to refer to a very nice publication of the previous author or the previous speaker, a study from Peter Koves. And there the objectives were to evaluate the exposure of discharge nuclear medicine patients and so to provide a detailed assessment that should give more information about the impact of on the individual radiation dose. And so in this study, they focused on two specific health workers. So they focused on the staff in critical wards, and they also evaluated the dose rates while performing ultrasonography. So the general concept of this dosimetry study was to measure the cumulated radiation dose over a long period of 6 months in order to increase the statistics, to measure the representative background, and also to record the patient data. So which nuclear medicine procedure was used to specify the ward, and also to in order to do this, the ward staff got kind of transformed hospital badge with an integrated dose meter, which led to a sustainable measurement without an extra inconvenience. And this was done for 70 staff members. This is an overview of the staff members, with also an overview of the number of patients that were in contact with every individual staff member over a period of 6 months. So these were the results of the radiation dose measurements for the background. And if we have a look at this, then you see that the mean background doses were at about 0.5 millisieverts over 6 months. And these were the results of the wards that were in contact with the nuclear medicine patients, showing no significant difference with the background measurements. They also focused on the radiation exposure from discharge nuclear medicine patients for ultrasonographers. In this evaluation, there were no individual measurements of the radiation dose, but dose meters were put on an apparatus, an ultrasound apparatus with a set of 3 pieces close to the bed, which led obviously to overestimation of the individual workload. And this was done for ultrasonography apparatus. These are the results. So on the left side, an overview of the number of patients over 6 months. So there was an evaluation of, or about 450 patients passed the ultrasonus apparatus, the 4 in total. And at the right side, you also see that there was no enhanced cumulated radiation exposure, which significantly differs from this from the background level. So this is a very nice example on how translational and observational research can be helpful both in risk communication and decision making. But there were some caveats for this study. So contaminations were not assessed in these studies, and also there was no information, or at least there were no patients involved that had undergone a therapeutic procedure in nuclear medicine. So this should have a separate attention. In this slide, these are the occupational radiation exposures in our own department at ASA Delta, showing some dose rates for lutetium PSMA and lutetium dotatate. So the radiation dose rate against the transportation packaging or the container when the product arrives at the department is less than 1 microsievert per hour. when you do the measurement against the vial with beta shielding, then at the site we have some dose rates of 10 microsieverts per hour, at the upper side about 35 microsieverts per hour. Behind the screen, and that's important, that's the place of the operator, the dose rate is below 1 microsievert per hour. And when also during the administration behind an automatic syringe pump, the dose is around only 1 to 2 microsieverts per hour. Immediately after the administration, we have dose rates at 1 meter from the patient of about 50 microsieverts per hour. And at 2 meters, it fastly degrades or decreases to 2 to 3 microsieverts per hour. The next day at discharge, so in Belgium, we have the obligation to keep the patients for an overnight stay when they are treated with beta emitters, such as lutetium-177. And but at discharge, these are is an evaluation after at least more than 200 treatments or administration, and we never found a dose rate of more than 20 microsieverts per hour on the next day or the next morning. So, and this is also just an overview of the dosimetry of the nurses that are treating or helping the patients that are hospitalized in the radionuclide therapy rooms. And also there you see an overview and the cumulative dose at the left side, not exceeding the highest dose is about 0.2 millisievert per year. And so it's about 1% of the legal dose limit per year for occupational exposure. And then in order to conclude a few slides, and I think that's from our point of view or from our perspective, I think the most important part is it's all about risk communication. And obviously there are different approaches of this communication, and it's a kind of patient-tailored communication where you have three main aspects. On the one hand, you have the patient population, and you have to take into account the patient background, also the education of the patients, the general misinformation that exists about radiation, and also the age. And you see a nice representation of a very nice initiative, which is a book on covering the adventures of Tim and Sunny, which is really dedicated to communication to children and to inform them about all aspects of nuclear medicine. So there is a reference underneath. And then the clinical context, you always have to find a balance between the risk and the benefit. But there's also the risk of not performing the examination or the therapy, obviously. On the other hand, you have the radiation risk, which should also be taken in account. And there are three big different approaches in order to communicate. And when it comes to the protection of the public, we generally use the non-quantitative approach, where we will compare the risk with other radiological tests, and especially with the natural background radiation, while when we perform diagnostic examinations in a patient and we evaluate the medical exposure, we will rather use the relative risks, which is the comparison, for example, of the cancer risk associated with radiation exposure to the natural risk of cancer, which is shown on the left side, or the lifetime risk, which where an example is the risk associated with radiation from medical imaging to other lifetime risks, like shown on the right side. So this is the last slide, just to show you that there is already quite some information available when it comes to radionuclide therapy. So there are very practical EANM technologies guides, which are freely available through the EANM website. You have the publications on radionuclide therapy procedures, such as the joint procedure on treatment with lutetium PSMA, and also some guidance on how to set up a theranostic center. On the other hand, you have a global overview, and that's also this book is also available as an app, and with a complete overview of all nuclear medicine procedures, including also topics on radiation protection. And so, as a conclusion, I would like to stress the need for radiation protection related research, but as well as for the need for multidisciplinary collaboration in order to establish the right balance between regulations and medical needs without hampering the introduction of these new therapies in clinical practice. Thank you.
Thank you very much, Christoph, for the comprehensive presentation. You also mentioned on the very important topics about risk communications. I think we did not yet include this topic, but we will talk about later on. Thank you again. The next speaker is Lara Stulens. Lara is the head of the research group on radiation application at SCK-CEM in Mol. She is member of the scientific committee at Medical. She, of course, is the secretary of the pilot group and also the member of the pilot group, and as well in the working group 6 in EURADOS. Lara, the floor is yours, please.
Okay. Thank you very much, and I hope you can see my presentation. Good afternoon, everybody. So I will indeed finish the webinar by showing what we did together with different partners, different working groups, and the EANM Radiation Protection Committee.
Lara, your screen is frozen, I think. Maybe you can restart. Strange. I tried to contact Lara.
Yeah, looks like Lara has connection problems. We will try to get in touch with her.
Yes.
Sorry for everyone.
Yes. Please a little bit patience. We contact Lara and come back.
On developing a computation.
Lara, you are frozen in the presentation.
Okay. Sorry, I understood nobody could hear me. Can you hear me now?
Yes, Lara, you have to start again. Thank you.
Yes, we can hear you now.
Start again. I don't know what
You can
happened.
start again, please.
I was just
No
talking.
problem. Yes, yes.
Okay, wait. I'm really sorry. Okay, tell me, can you see my presentation?
Yes.
Yes.
Yes. Can you hear me?
Yes.
Yes.
Okay, I'm sorry for the delay. I will start over again. I had no idea. So good afternoon, everybody. I will be a little bit quicker, but so I will explain on the work we did on the computational approach to assess external dose rates from nuclear medicine patients. And as we could already hear, the current risk assessments for people coming closer to nuclear medicine patients, they are based on ambient dose rate measurements in a single position from the patient at a specific distance, and they could be carried out at several time points after administration of the radiopharmaceutical to actually estimate the whole body retention of that patient. Now, once this measured dose rate is below a specific patient release criteria, then the patient can go home, but that could then result in an exposure of the public, a caregiver of the patient, or when he's back home, a family member. Now, this approach means that a series of simplifications are made for determining both the radiation emitted by the patient, but also for the dose which is received by a person in proximity of that nuclear medicine patient. Because if we look at the nuclear medicine patient as a radioactive source, then it is actually not a point. It's a physically large source. We also are dealing with patient-specific biokinetics, which means that the activity distribution in the patient changes over time, and this can be both isotopic and pharmaceutical specific. So we hypothesized that using a combination of a point measurement and one unique effective half-life of the radiopharmaceutical could actually result in errors, specifically when we look at very close contact scenarios. So that is why we started the work on developing a computational approach to assess better these external dose rates from nuclear medicine patients. And it's a joint work from different EURADOS working groups and from the EANM Radiation Protection Committee, because we actually needed different types of expertise for this work. So this computational approach exists of different modules. One of them is that if we want to better assess these external dose rates, we wanted to better and more realistically simulate, let's say, close contact scenarios. So we needed computational models that are flexible, so that we can change the posture both of the patient and of the caregiver or the family member or the general public. We also want to take into account more in detail the biokinetic models, so how the activity changes over time and within the different source regions of the patient. And all of that actually is so what we use as to describe the nuclear medicine patient. And then the goal is to use this input data for Monte Carlo modeling, then to actually come up with a better description of the exposure of the caregiver or the family member, where also that person is represented by a computational model, so not a point anymore, which then results in this type of curves you can see here, which are actually organ-absorbed dose rate curves over time, so post-administration of that target person. Okay. Now, what are the different tools we are using here? So for the biokinetic models, we have been using the ICRP reference biokinetic models, but we have also been using more revised ICRP models based on patient data when they were available. For the flexible phantoms, we have been using ICRP reference phantoms, but also some in-house developed phantoms from partners within the study, including also the software, of course, to change the posture of these computational models. And then to do the Monte Carlo modeling, we had different partners involved, and several codes have been used: MCMPX, FITS, GATE or GEANT, and the EEE4. Now, how did we came up with these organ absorbed dose rate curves? So let me explain. So what we are interested in, so the curves you can see here in orange and in blue, is the total organ-absorbed dose rate for the target person. But because we know that the effect of all the different source regions for a certain radiopharmaceutical and the effect of the source regions to the total absorbed dose is additive, we actually performed all the Monte Carlo simulations for each source region individually. And that's what you can see here. So we calculated with the Monte Carlo codes and the flexible phantoms, the organ absorbed dose rate in the target, but per activity in a specific source region. And then we multiply this with the information from the biokinetic models, so the fractional activity on a certain time t in a source region. We do this for all the source regions separately. We sum them all up. We multiply with the administered activity to the patient to get then these type of curves, which are the organ absorbed dose rates for a specific considered target. Now, of course, we wanted to benchmark our computational framework, which meant that we compared the different Monte Carlo codes of the different partners, but we also made a comparison against experimental dose rate data that we obtained from nuclear medicine patients. We did that for specific clinical radiopharmaceuticals and procedures. Of course, for the benchmarking, we looked more at simplified geometries, like two computational models just facing each other, or simulating what is measured in clinical practice is here just the dose, the ambient dose equivalent rate, H*10, at a certain distance, let's say 1 meter from the nuclear medicine patient. So this was just for benchmarking purposes. Let me just show you a few of the results that we obtained. Here you can see the comparison of different Monte Carlo codes for two computational models facing each other at a 1 meter distance. And you can see here the results, the final effective dose rate per administered activity from the different codes. And in general, we found a variation between the codes, which is always less than 8% for all source regions and all radiopharmaceuticals considered. So we were quite happy with that variation between the codes. We also compared against experimental dose rate data from these nuclear medicine patients. So this is more this setup that we considered. And here you can see each dot actually is a dose rate measured at 1 meter from the patient. In this case, it was for fluorine 18. These measurements were done by different partners in nuclear medicine departments. We simulated this setup as well by different codes, and then the big gray line is the average for this specific scenario from all the different codes involved. And you can see the dotted lines are the 95% confidence intervals on the simulated data, and you can see that measurement data fall within these confidence intervals. And here you can see a similar graph where we compared measurements for lutetium, both for DOTA-Tate and PSMA, against what we simulated for this specific radiopharmaceutical, again, for this same setup, as you can see here above. Now, we have many more benchmarking data, but it was too much to show in one presentation. So if you're really interested, we invite you to read the publication that we have written and published on the benchmarking of this computational framework. But so, of course, the next step, we have confidence in our computational framework, so we want to apply it, of course. And the first thing we did is to look at what is the impact of all the different postures that we can come up with, let's say, between a nuclear medicine patient and a target, which could be a family member, a caregiver, a person of the public. And the first thing we did was actually categorize the different contact scenarios. And here, what I will show you here and in the rest of the presentation is specifically for a moderate child configuration, but we could do it actually for any configuration. So we defined some like close contact scenarios, like really physical scenarios where there is direct contact between, in this case, a mother, which is a nuclear medicine patient, going back home and is in contact with one of her children. We also defined some other postures. This is just a snapshot where the distance is around 30 to 50 centimeters. There are many more where we change orientation and so on. And then other configurations where we have, again, a little bit of a larger distance between both computational models. Okay, so what is the impact of these postures on the external dose rate. To do that, well, I come back to what I explained before on the concept of the study. So let's assume one specific scenario where we say that the mother is a patient which is treated for hyperthyroidism, for example. So she's treated with iodine 131, and she receives an administered activity of 400 megabecquerel. So it means that we have to calculate, and we are interested in what is the external dose rate to the child. Okay, so we have run the simulations as I explained before, not for all the different postures yet that we defined, but just for a few of them, and I demonstrate them here. So for all these postures, we have calculated data from the framework. So I will show them for these postures. And we combine them, of course, with iodine 131 biokinetics. We selected the ones from ICRP 128. And these, of course, define the source regions that we used in our Monte Carlo simulations. And as I demonstrated before, then we calculated with the same formula, in this case, the effective dose rate to the child, considering all these different postures. The result you can see here. So these are these effective dose rates to the child from all these individual postures that we have defined. And you can see color-coded that the ones where there is really close contact, you see indeed that the effective dose rates are higher. And then the blue green curves are the ones that I showed here in the green area. So, okay, what can we do with these specific curves? Let's say, for example, that we are interested in what is the effective dose to the child after being in contact with his or her mother for 14 days after administration. And then we, of course, assume, we have to make some hypotheses, and we assume that for every 24 hours, they have 1 hour of physical contact, like really close contact, like you can see here in the orange pictures, and per 24 hours, 6 hours of more, like we call a social contact. So there is no direct contact, but it's pretty close. Okay. Of course, we have a lot of course to work with, so that's a little bit difficult. Sorry. So we decided that for each of the two categories, distance categories, we chose the curve that gives the highest effective dose to the child and the one that gives the lowest effective dose to the child, so that we can come up with a dose range, let's say, for this specific scenario. Okay, but another question we have to ask ourselves is, like, from what point on will there be contact between the child and the mother? So from what point on will that mother leave the hospital, and then the contact with the child could actually occur? So for this, actually, we simulated also the H*10 curve, so just a dose rate at 1 meter distance from the mother, and which is represented by the black curve. And then we compared, or we looked for the time when this ambient dose rate equivalent reaches a value of 20 microsievert per hour. That was just a selection of a typical release criteria. As you could see from the first presentation, it's not a harmonized criteria. And we can see here from this curve that actually the 20 microsievert per hour is already reached after 2 minutes. So it means, which is also normal with this level of activity which is administered, the patient can actually leave the hospital immediately after the treatment. So we could start calculating this specific scenario from the start, from the very early time points. And this is what we did. So for each 24 hours here, we calculate, or we assume 1 hour physical contact and 6 hours social contact. So we use these curves, we add them all up, and then we can see an example here, just for a demonstration, that after 14 or 15 days, the dose range given to the child from being in contact with her mother in these specific scenarios ranges from 0.8 to almost 3 millisievert. And then we can, of course, play with the data. This was the scenario that I just showed you now. What if we increase the amount of activity given to the mother treated for the same therapy? And then we can see that the H*10 at 1 meter reaches the 20 microsievert per hour after 24 hours, which means that the mother would stay at least 24 hours in the hospital before going home. So we don't count day 1 in our calculated effective dose curves. And then you can see the same calculations are done, and we reach still, although they stay in the hospital for 24 hours, we reached ranges of effective dose to the child going from 2 to 7 millisievert. Another scenario, just to show as a demonstration, what if the mother is not treated for hyperthyroidism but for thyroid cancer? It means that we changed our biokinetics. We used the biokinetic data that was available from thyroid cancer patients. We of course increased the administered activity in this case, and then we could see that the 20 microsievert per hour at 1 meter distance from that patient was reached after a little bit more than 2 days. So we started counting the external dose rate to the child from day 3 on, and we reached a total after 14 days going ranging from 0.7 to 1.6 millisieverts. So here, what I intended to do was just to demonstrate what we can do with our computational framework. So we are not done yet with calculating these organ-absorbed dose rates or effective dose rates to a target for all the postures we already defined, but I demonstrated it for the ones that we already did. So in summary, we have a benchmark computational framework, and it exists of 2 separate modules. One is the Monte Carlo-based calculation of the external dose rate per unit of administered activity, and the other module is the description of the time activity distribution for each source region, depending on the biokinetic model that we use for the respective radiopharmaceutical of interest. The fact that the modules are separate are nice because we can do the Monte Carlo, and if we want to change biokinetics because we just have better models or better data, we don't have to repeat actually the Monte Carlo anymore. We can do this for any posture and any spatial arrangement between a nuclear medicine patient and a member of the defined critical groups, the general public, staff members even, caregivers, family members, and so on. And we can do it for any radiopharmaceutical. But to mention is that one of the current limiting factors is to actually have appropriate biokinetic models for nuclear medicine patients. This was a little bit out of scope of this presentation, but it certainly is a work in progress. So what is there for the future? We will continue to calculate these organ dose, the organ absorbed dose rates for targets for all the different postures that we defined. To at the end come up with a tool of these organ absorbed dose rates per unit of administered activity for a large variety of close contact configurations for a range of radiopharmaceuticals, both diagnostic and therapeutic. And maybe also there will be an opportunity to make it more patient specific by including, for example, some individualized features like whole body retention measurements performed on the patient itself. Now, what can we do with all this data? Like mentioned in the first presentation of Peter Govens, we can actually use it to evaluate or to provide the necessary data to evaluate what is the expected exposure to caregivers or to the public for specific scenarios as part of these risk assessment studies. And then with the choice of appropriate dose constraints, which is another topic that would facilitate then the setting of hospital release criteria and patient restrictions. So thank you for your attention, and I'm willing to answer your questions.
Thank you very much, Lara, for the outstanding presentation. Now I think we are in the sessions questions answer. I would invite the speakers to open your camera maybe, and also Francesco to read out the questions.
Yes, thank you, Weibo.
Yes.
I would answer the question in the reverse order they were asked. So we start from the last question that it comes from Valentin Bonvin. Don't know if I pronounce it correctly. Did you calculate the effective dose for different scenarios in the case of lutetium PSMA therapies? This tool seems to give the right answer to harmonize the release of patient.
I guess that's a question for me. We started with the lutetium data. We benchmarked our framework with lutetium data. So for all the postures that I showed within the presentation, we will definitely do it for lutetium as well. But I think we still need to do some work as well on the biokinetics. There is some data already. But yeah, once we defined both parts of our framework, we can definitely do it for lutetium. And I think it will be one of the next steps in the collaboration.
Okay, thank you, Lara. Second question comes from Naomi McElroy, and she asks, but for the thyroid cancer, are you including excretion or just physical decay?
For any radiopharmaceutical or therapy that we consider, we take into the biokinetic model. So yes, we include excretion and how the activity changes over time, not just the physical decay.
Okay, thank you. Another question should be for Peter. Okay. When evaluate, it comes from Sakai Kinase. When evaluating the dose of a representative person, do you specifically evaluate the dose of 1-year-old, 10-year-old, and adult?
It can all be done. It's part like Lara said in her presentation. It's part of the potential exposure scenarios that make part of the contact with a 1-year-old or a 10-year-old and an adult. That could make up the release or the patient instructions. And true in setting, for example, or evaluating those to a representative person, we need to know that potential scenarios, especially for a member of the public. So yes, it can be done.
Okay, thank you, Peter. There is another question for you coming from Christian Luca on about, do you establish those constraints and those limits within the EU for medical exposure? What proportion constitutes deterministic effects, short-term or acute, and what proportion constitutes stochastic effects, long-term or random?
Yeah. First of all, there are no limits for medical exposure, of course, and there are only constraints for medical exposure if it's for the comforters and carers. And in that case, it's only stochastic effects that are considered. It's the optimization of the radioprotection in the framework of stochastic effects, in the framework of limiting stochastic effects.
Thank you, Peter. Then there is a question coming from Gila Karimi Diba. I think Peter already answered to this question because she asked more about those constraints.
True.
Stressing the difference between those constraints for radiation protection purposes and for the patients and caregivers.
In fact, those constraints in the framework of medical exposure are only for comforters and carers, because it's more or less the same. It's also an optimization of protection for really medical exposure of patients. It's called reference levels, not dose constraints. And to answer the other question, I think there's one above. Why is the exposure of comforters and carers in fact considered as medical exposure? That is because already for years it was recommended. It's because it occurs in the context of a healthcare setting where, in fact, the radiation is intentionally used for diagnostic or therapeutic procedure. So, in fact, the comforters and carers are not direct subjects of the medical exposure, but their exposure, in fact, is necessary. It's a necessary part of providing care and support to the patient. So, in fact, carers and comforters, they are defined as individuals knowingly and willingly incurring an exposure to ionizing radiation by helping, in fact, as sometimes in part of their occupation, in the support and the comfort of individuals that have undergone a medical exposure. I hope it's satisfying as an answer.
Yes, looks like, yeah. There is another question for clarification about the previous questions. Thanks for clarifying that biokinetics is used. I was considering the issue of metastatic disease, where we tend to see higher uptake in the thyroid cancer patient, and also if a patient cannot stop a medication such as lithium, it can take longer for the activity to reduce within the patient. Do you want to say something else, Lara?
I'm unmute myself. What I can say, okay, there are also some people here in the audience who are much more experts in biokinetic modeling than I am. I know that within the meetings we have on the topic, this is something that we discuss a lot. The fact that for patients, the biokinetics are so different, and indeed thyroid cancer patients, what is the uptake in the thyroid, and for the moment we don't have maybe the perfect biokinetic model yet, but this is work in progress, and I think more and more patient data might become available. And then, as I said at the end of the presentation, we don't have to restart all the work and all the curves we calculated, because the effect of the biokinetic model, we can just change it separately from the Monte Carlo. So yes, we are aware of the difficulties in the biokinetic data of different patients and different types of therapy.
Okay, thank you, Lara. We have the last question coming from Anne McCann. Is more or less addressed to all speakers. Does anyone have any thoughts or experience of patients staying in a hotel after therapeutic 177 lutetium administration? For example, a patient who lives far away from the hospital and has to return the following day for scan. How do you address the risk of bodily fluid contamination in the hotel? Even with good hygiene, there may be some contamination.
Well, that's the problem of the heterogeneity in the regulations, I think. So, for example, in Belgium, we have the obligation to keep the patients for at least one night in the hospital for 24 hours, and then most of the urinary excretion already happened. And to be honest, as a clinician, I'm not that much worried about the dose rates, but to avoid contamination is another topic, and that's why I stressed in the last part of my presentation the need for communication and to adapt your communication depending on the particular situation of the patient. So if I would have a patient that is telling me that he's going to a hotel, I would more stress to reduce or to avoid the risk of contamination for sure. So, but this is what this is just clinical reality, and it's very important to judge upfront and to see your patient yourself before giving the treatment, to judge the situation at home or the specific situation of each individual patient, and to adapt your communication likewise. Yeah.
Maybe I can add from a physicist point of view. There is a recent publication from a Dutch group, and they showed the activity concentration in the urine over time after the application of PSMA targeted therapies, and they showed that in the first 6 to 8 hours, even a small drop of urine might lead to an overexposure to more than 50 millisievert to the extremities. So that's exactly what Christoph said. If you know you have a patient that suffers from incontinentia or something like that, you have to focus the communication on this aspect.
Yeah, thank you very much for the comments. Yes, we will take this to look at the paper. Yes.
No more questions. Thank you.
Yes, there is a one, I think. The last just come from Olga, you know. When do you...
Yes, 1
Yes.
minute ago, yeah.
Yeah.
When do you believe there is going to be a compliance of directions through all the country members? What can we do to help? No, good question.
Yeah, that's a really good question. If I could look into the future, in fact, we don't know, because it could be political choice also, but within SimpleRad, we proposed some remedies that could help to speed that compliance or harmonization. But it will not be a very simple task to do that, because we need competent authorities certainly to accomplish this harmonization. But I think not only this topic on external dose rates at close contacts, but also other initiatives could help to go for a higher harmonization level in the future. But at this moment, I don't know when it will happen, but it will not for tomorrow.
I would add we can join the EURADOS community that can help a lot in networking between scientists to do something from our own. Sorry for saying that.
I think there's no questions anymore to be answered. I think, Francesco, we answered all the questions, right? Okay.
Yeah.
Okay, then I will close our webinar. Thank you very much again for the listeners and also the speakers. Excellent. And also Jens Kort from the EAM. And also Francesco and the casting and Agnieszka for the organize this webinar. Thank you very much. See you next time. Bye-bye.
See you.